molecular and cell biology in the Division of Immunology and Pathogenesis. And my project for this summer was studying the role of non-hematopoietic cells in tuning natural killer cell responsiveness. So there are two really important white blood cells that you need to know about for today's discussion. And they are the natural killer cell, which is this guy right here, and the T cell. And I've sort of dressed them up in this army here because they are the two more important cells in killing dangerous cells in your body, such as tumor cells or viral cells, such as this guy right here. So they are a very important part of our body. And the way that they're able to kill these dangerous cells is through the utilization of this one molecule called the major histocompatibility complex class 1, which are these guys. And the MHC molecule is on the outer surface of most every cell in your body. And the way it works is the MHC molecule, when it's on the outside of a happy and healthy cell, it will inhibit the natural killer cell and stop the binding of the T cell, therefore allowing the happy and healthy cell to move along on its way and stay alive. Now, when it's on a dangerous cell, such as this guy, it still inhibits the natural killer cell. However, it activates the binding of the T cell, and therefore the T cell will bind to this MHC complex and kill this dangerous cell. So this is the main concept that you need to know for this discussion, that MHC molecules let the T cells and natural killer cells know if the cell that they're on is dangerous. So dangerous cells have actually evolved over time and have become incredibly intelligent in that they're able to hide these MHC molecules on their outer surface so that they're no longer there. This then creates a stealth dangerous cell and makes it so that the T cell is no longer able to see the cell and just sort of moves along on its way as if it's a happy and healthy cell. However, our bodies are also incredibly intelligent in that the natural killer cells, since before they were inhibited by this MHC molecule, they're no longer inhibited and actually activated by its lack of presence. Therefore, the natural killer cell will attack this stealth dangerous cell and cause it to die. This is called responsiveness. So responsiveness is the ability of this natural killer cell to attack these dangerous cells and kill them effectively. So biologists started to wonder when they found this interaction, what would happen if the natural killer cell was placed in an environment that was completely MHC negative. So as you can see, all of these cells do not have those small MHC molecules on their outer surface. So they bred mice that had several different strains of mice that were mostly MHC negative and wanted to see what phenotypes were presented. And they found that in these environments, the natural killer, or the mice, were relatively normal, except that their natural killer cells were sort of sleepy and had a very low responsiveness. And our theory is that this is because the natural killer cells, due to the fact that they're trying to attack each and every cell, become exhausted and tired due to this large expenditure of energy and just stop responding to any stimulation. And we found in the past that if you take these exact same natural killer cells and transfer them into an MHC positive environment, they'll become happy and healthy and fully responsive again. And we'll be able to kill these dangerous cells through the activation of the lack of the MHC complex. So through this ability for the natural killer cell to change responsiveness due to the environment, we sort of based our entire research. So we found that MHC interactions are completely necessary for the natural killer cells to be functional at all. So they need a little bit of interaction with those in order to recognize that there aren't any on some of those cells. And that the responsiveness of the natural killer cells can easily change with a changing MHC environment. So from here, my question for the summer was if there was a specific cell type that was determining this change in natural killer cells. So if there was a specific type of cell in the MHC negative or positive environment that caused the natural killer cell to become less or more responsive. So we broke up cells into two broad categories. These categories are hematopoietic cells and non-hematopoietic cells. Now, the hematopoietic cells are blood cells, cells that are in your immune system, things such as this. And the non-hematopoietic cells are stromal cells, which are cells such as fibroblasts and osteoblasts, which are in your bones, lungs, and skin. In a past in vivo experiment, we found indications that the non-hematopoietic environment was actually the environment that was sort of tuning this natural killer cell responsiveness, which was the important environment. And we wanted to see if we could set up an in vitro experiment to see if we could further analyze this, find a specific part of the non-hematopoietic environment that was important, and then reapply this in vivo. So we created this general experimental layout. First, what we wanted to do was we wanted to create a completely non-hematopoietic environment. This was the most important part of our experiment because in order to test if it was only the non-hematopoietic cells, we had to get rid of everything else. Next, we wanted to determine an extraction procedure for the natural killer cells, which I will explain later. And once we were able to extract those natural killer cells, we wanted to plate them on both an MHC positive and then an MHC negative environment of the non-hematopoietic cells we established earlier to see the changes in responsiveness for natural killer cells from the same mass. So the first part of our experimental layout was establishing the stromal environment. So in past literature, we found that if you take bone marrow and you irradiate it, the hematopoietic cells will die off and you'll create a completely stromal environment due to the increased vulnerability of hematopoietic cells to irradiation. So we took a little mouse and took his bone and we extracted the bone marrow and plated it. We left it for 3 weeks for it to grow and proliferate and gave it one large dose of irradiation. We waited 1 week for the hematopoietic cells to die out, and then we tested it for the amount of hematopoietic cells in culture. And we found through the cell surface marker CD45 that is primarily on hematopoietic cells that there was still between 30 and 40% of hematopoietic cells in culture in both types of MHC environments. So we figured that since one dose of irradiation was able to reduce the amount of hematopoietic cells so much from the bone marrow that multiple irradiations would do even more. So we continued and irradiated the bone marrow even more. We created an assay in which we took bone marrow just the same way and either pre-irradiated it or did not pre-irradiate it before plating. Then we irradiated it each week, testing it each week the amount of hematopoietic cells with the CD45 marker. On week 1, we found a relatively low amount of hematopoietic cells in culture, especially in the not pre-irradiated one, which increased as week 2 and week 3 went on. Finally, in week 4, we found relatively low amounts of the hematopoietic cells, which made this the ideal week to establish our non-hematopoietic environment. So this was the accomplishment of our first step in our experimental procedure. So this was the establishment of the stromal environment. And we wanted to know if we could establish stromal cells in a different environment, to have 2 environments to compare it between. So we took lungs and digested them, breaking them into tiny pieces, and grew what the literature suggested was fibroblasts out of them, and found a similar result of almost completely negative CD45 cells in culture, which made it our second success in the first part of our experiment. So we moved on to the second part of our experiment, was determining a way that natural killer cells were able to stay in culture for long periods of time. Now it's well known that natural killer cells die when you leave them in long cultures like we would have to for this experiment due to a lack of growth factor. So we wanted to find an amount of IL-15, which is a major growth factor for natural killer cells, so that they would be able to survive throughout our entire experiment. So we negatively selected for natural killer cells, which means we used magnetic beads to get rid of all of the random cells from the spleen, and then we plated them for different amounts of days and different amounts of IL-15, and found that on day 1, the different amounts of IL-15 didn't really matter because they didn't have time to die yet. However, that on day 5 and 7, there was significant difference between 10 nanograms per milliliter of IL-15 and 1 nanogram per milliliter of IL-15, therefore indicating that this was the best way to determine or to culture natural killer cells for a long period of time. So in conclusion, throughout the summer, we created a protocol in order to establish this non-hematopoietic environment and the best way to culture the natural killer cells. And in the future, we want to do this co-culture with the stromal cells and the natural killer cells on top to see this difference in responsiveness due to the MHC environments. And in order to do this, we have to determine the best way to purify the natural killer cells, which is either using the whole spleen, which is relatively unpure but also very easy, using negative selection like we did before with the IL-15, which is much more pure, or using green fluorescent protein in the natural killer cells of mice and selectively sorting for those fluorescent natural killer cells, which is the most pure type of selection that we could do. Once we determine the best way, we're going to take those isolated natural killer cells and plate them on the fibroblasts or bone marrow-derived stromal cells that we established earlier this summer and test the different responsiveness due to them. At this time, I'd like to thank my PI, Dr. David Raulet, the entire Raulet lab, and my mentor, Dr. Michele Arlino, for all your help over the last year, and especially the SURF program and the Rose Hills experience for giving me the opportunity.